Let's have a look at how starch is digested. Now, it's important to understand that glucose is the main energy source for most or almost all most living beings, even for plants. Of course, plants obtain their energy from the sun, but they use this to make glucose, and then this glucose that they make is used for cell respiration. And it's important because glucose can not be found in nature in pure form, but it's mostly bound together, connected into long chains to form starch. So if you want to understand how this starch digestion is possible, we have to first understand a little bit how glucose looks like. And as you might already know, the chemical formula for glucose is C6H12O6, 6 carbons, 12 hydrogens, 6 oxygens. Now, if you look at this structural formula over here, you're going to see that many of the carbons are missing or seem to be missing. I see one carbon over here. Where are the others? They are implied. They are not written. Every corner over here represents a carbon, but they simply didn't draw it because it's clear anyway. What else should it be besides carbon? So every corner is a carbon. The same for the hydrogens. They are also implied. They are also not all shown because if you don't write it, it's got to be a hydrogen. And now, if you count all the hydrogens, oxygens, and carbons together, you should get the formula chemical formula for glucose. So, but there's another thing that I need to explain over here. If you look at glucose over here, you see it's a circle. And there's an oxygen over here in the circle, and we go clockwise and we give each carbon a number. So this is carbon number 1, carbon number 2, number 3, number 4, number 5, and the carbon outside of the circle is carbon number 6. So that's basically their names. And for whatever reason, chemists many years ago, they decided if you have a ring shaped with an oxygen over here, then we start counting the carbons clockwise right after the first carbon, right after the oxygen. This is simply a convention that they used and invented to make communication within the scientific community easier. So, and this numbering we have to know as well, because now I'm going to show you the formula of starch. And don't be confused, it's simpler than it appears. So, starch are long chains of glucose, and however there are two types of starch and they have a name: amylose and amylopectin. And amylose looks like this, and each one of these structures here is a glucose molecule. And look that over here, carbon number 1 of this glucose over here is connected to carbon number 1, 2, 3, to carbon number 4 of the next one, and the same over here, carbon number 1 to carbon number 4. So amylose has 1,4 bonds. Okay, and it's a long chain. If this is for you too difficult to remember, simply replace every glucose molecule with a circle. That's a glucose, that's another one, that's another one, that's another one, and so on. Actually, to be very honest, it spirals. You don't have to know that. Just remember that in amylose, you have 1,4 bonds. Now, what about amylopectin? Do we also have 1,4 bonds over here? Yes, of course. We have a 1,4 chemical bond over here. But what else do we have is we have over here a branch for this carbon number 5 over here. This is carbon number 6. So you also have 1,6 bonds. So it has 1,4 and 1,6 bonds. And this is pretty important for understanding when you want to understand starch digestion, because not all enzymes are able to break all of these bonds. If this is too difficult for you to remember, then simply remember again a nice little circle diagram over here. So that's basically, I'm still drawing my amylose, but there are some occasional 1,6 bonds, and you know what, over here as well. It's like almost like a network. You get the idea? Okay, so that is amylopectin. And we continue, okay? And maybe over here as well. You get the idea. So, so that's basically important for understanding, because what happens now is that we have an enzyme called amylase. And if this is amylose over here, it's able to break it down. And this amylase is released by the salivary glands in the mouth and also by the pancreas. And what this amylase does is the following. It chops these bonds over here. And we always have two glucose molecules. And two glucose molecules joined by these 1,4 links has a name, and that's referred to as maltose. So we have a maltose molecule over here, we have a maltose molecule over here and over here. And amylase has no problems doing that, and it will do that. But it's only able to split these 1,4 bonds here. And now we move on to the next one. And inside cells, what happens is that the maltase, that's another enzyme, is now able to further break down one maltose to 2 glucose. So the maltase is able to chop the last 2 glucose molecules apart. Now, when you digest amylopectin, we have some parts left over which have these 1,6 links, because remember, amylopectin has the 1,6 connection. So everything else was digested by the amylase, but these things are left over, and maltase cannot break this down. So what we need is we need dextrinase, and dextrinase will split it up over here, and then we have again 2 glucose molecules being formed if you use this here as a dextrin as a starting place. So that is basically the importance here. Why so many details relating to starch digestion? Well, because again, starch is the main energy source for most living things, and at least human beings. If you look at all cultures, will use starch as their primary energy source.